Hello once again. We are now in the screencast for the family Spirillaceae. Under the family Spirillaceae, there are two important genera that are involved in human diseases. This would include the Campylobacter and Helicobacter. There are many species of Campylobacter. Among those that are noteworthy to mention are Campylobacter jejuni, subspecies jejuni, okay, Campylobacter fetus, and others. So these organisms are microaerophilic, which means that they would only need small amount of oxygen for them to grow. And one of the most important hallmark is the seagull wing appearance, morphology of these organisms. So they are positive for oxidase test and catalase test, and the motility is described to be as the darting motility. These species or these organisms are non-fermentative, which means that they are not able to ferment any sugar. So the term campylo would refer to organisms that is curved or comma shaped. Okay, since there are two comma shaped organisms, it appears like seagull or the wing of a seagull. Okay, so Wartin starry medium. Okay, so let me go back to the previous slide. This is a Wartin starry stain. So this is a very sensitive stain. Okay, Wartin starry would be a very sensitive stain for Campylobacter. Okay, so the organisms are microaerophilic, and if we're going to collect the specimen, you have to use a transport medium, and that is known as the Carey-Blair transport medium. Campylobacter or the Blazer medium is also a selective medium for Campylobacter because of the presence of vancomycin, okay, trimethoprim, and amphotericin B. Then, aside from that, you could possibly use the Butzler and Skiro medium, medium B. Campylobacter is foodborne and would also be considered as a zoonotic infections. So, as you can see, there are over 13 species of Campylobacter, but the most commonly associated with human infections would include the Campylobacter jejuni and Campylobacter coli. And of course, to be left out would be Campylobacter fetus. Campylobacter are zoonotic infections, which means that you could possibly get it by interaction with dogs, cats, especially chickens and cattle. Okay, so transmission to human is primarily by oral or fecal route. So since there's small comma-shaped motile and they would require microaerophilic for growth, it is also noteworthy to mention that the organisms would best grow at 42 degrees centigrade. So the antigenic structure is almost similar to the Vibrio and other Enterobacteriaceae, but there are many O antigens and H flagellar antigens. Now, this is an example of TSI. So it is interpreted as K over K because of red over red. Why is that so? Because the organisms are non-fermentative, which means that they're not able to ferment any sugar. So Campylobacter jejuni, coli, and Larry. These are similar species that are implicated in diarrheal illness worldwide. So the diarrhea is almost accompanied by mild abdominal pain, characterized by cramps. Bloody diarrhea may follow with fever, chills, and sometimes could also cause nausea and vomiting. So the diarrhea would be self-limiting, which could last for about 2 to 6 days, but individuals could remain carriers for several months. Campylobacter fetus is also isolated from blood cultures, and this type of organism should mostly infect individuals who are immunocompromised or elderly patients. So in veterinary point of view, this type of organism is known to cause abortions in cattle, sheep, and in pigs. So what would happen once you have ingested the bacteria? So it will reach the ileum near the colon, will adhere and burrow to the mucosa. So in other words, it is invasive. And that is the reason why the watery diarrhea is said to be bloody. Although a variety of toxins have been isolated, the precise nature of pathogenesis is not understood. Therefore, the severity of the disease may depend on the bacterial load on ingestion, which means that the more bacteria that you ingest, then it is highly or directly proportional to the severity of the disease. Or achlorhydria, or the lack of gastric acid, could also contribute. Debilitation, or a weak status, or being immunocompromised. So if you are a healthy individual, the diarrhea is usually self-limiting. Okay, then another group of organisms would be the Helicobacter. Okay, recently isolated in 1982. Important species include Helicobacter pylori, Synedi, and Feniliae. So humans are probably the main hosts, but transmission routes have not been established. We're able to isolate these organisms in mucous gels that coats the stomach epithelium. Okay, so these are the survey of some important species of the family Spirillaceae, or otherwise known as Campylobacteriaceae. And of course, you have to remember the clinical significance. That's the reason why you have to review the table 20-6 in your Mahon book. Okay, so Campylobacter jejuni and fetus are commonly implicated or isolated species of Campylobacter. That's the reason why it's very noteworthy to differentiate them. So Campylobacter fetus could grow at 25, both could grow at 37, but only Campylobacter jejuni could grow at 42 degrees centigrade. It is also only the Campylobacter jejuni that could hydrolyze hyperate, and Campylobacter jejuni is susceptible to nalidixic acid. On the other hand, Campylobacter fetus is resistant to nalidixic acid, whereas it is somehow sensitive to cephalotin, which is resistant to, wherein Campylobacter jejuni is resistant to, or resistant with. So I did mention a while ago about Helicobacter pylori. Very unique type of organisms, because these type of organisms could produce a special enzyme that could neutralize gastric acid. So it is said that about 20 to 40% of adults in the United States have Helicobacter pylori colonization. So we do not have data here in the Philippines, but nevertheless, once Helicobacter pylori has colonized your stomach, it would result in 2 chronic superficial gastritis. So there is also a possibility that person-to-person transmission of Helicobacter pylori could occur. It doesn't invade the tissue, but they are capable in living in the mucus, covering the surface of the stomach. How could that be? The stomach is such a hostile environment for many bacteria, but not for Helicobacter pylori, because of their capability of producing the urease enzyme. The urease could break down urea to form ammonia, and ammonia is alkaline. Since it is alkaline, therefore, it could neutralize the stomach pH. So, as a result, the body would produce such a low-grade inflammatory response, enough to damage tissue, resulting in ulcers. So that's the reason why it is associated with type B gastritis, which is of course dangerous because this could be implicated in gastric carcinoma or stomach cancer. So what could be used to presumptively identify Helicobacter, you may inoculate it in Christensen urea medium. Christensen urea medium is a medium to test for the presence of urease test. So a positive result would be a deep pink color that could possibly change within 2 hours. You may collect the specimen. Okay, so gastric biopsy could be very useful. Have it stained with Giemsa, Gram, silver stain, or the Wharton-Starry stain. Urea breath test is also being used as a screening test for Helicobacter. So the patient may possibly drink the 14C labeled urea. The organism would degrade. The 14C labeled urea would be degraded by the urease activity by the organisms, releasing the 14C CO2. And this could be detected using scintillation counter. Okay, so what would you do? Okay, you may culture Campylobacter and Helicobacter using the Campy-blood agar. It is somewhat similar to Brucella agar base, except that you've added 10% sheep's RBC. Skiro medium could also be used. It is a blood agar base with lice horse RBC. The Butzler or the improved version of the bean media is actually consists of thioglycolate based agar media incorporated with 10% sheep's RBC. Or, of course, since the organism is microaerophilic, you have to be careful with the atmosphere that you'll be using. So it has to be about 5 to 10% oxygen with around 10% CO2 because the organism is also capnophilic. So this would be the different antimicrobial agents that you could possibly add in the culture media for Campylobacter species to make the organisms, to make the culture media selective. And of course, the different biochemical tests to differentiate species of Campylobacter, Archobacter, and Helicobacter. Okay, so I think we have already discussed Helicobacter pylori characteristics. Okay, so gastric biopsies are important by specimen. Then the urease enzyme is also important to neutralize the gastric acid. Okay, so this is an example of a Wharton-Starry stain. Okay, so you could see here that the organisms are able to invade the mucosa of the stomach. Virulence factors would include adherence factor, motility, and mucinase. The mucinase is an enzyme that could, of course, destroy the mucosal lining of the stomach. And this is the reason why there would be ulceration. So, Helicobacter pylori is associated with gastritis, gastric and duodenal ulcer because of the mucinase enzyme, and the type B gastritis, which could result into gastric cancer. Okay, so very unique since Helicobacter pylori can be found in the human stomach, normally considered to be hostile for most microbes. Okay, so that would be all, and thank you very much.